Dr. Melnick, can you give me some background on the mission of the National Toxicology Program, what your role was there, and your role specifically in the cell phone study?
Certainly. Just for a little background prior to the NTP question, the National Toxicology Program is headquartered at the National Institute of Environmental Health Sciences, located in Research Triangle Park. That is the NIEHS. That is one of the institutes of the National Institutes of Health. The National Toxicology Program comprises several federal government agencies that were brought together in the late 1970s for a single program. The agencies include the National Institute of Occupational Health and Safety, the National Cancer Institute, the NIEHS, as well as the Food and Drug Administration's research laboratories in Arkansas. So the National Toxicology Program was brought together to evaluate potential toxicity and carcinogenicity of substances that are of public health concern. The intention is to try to characterize the effects of environmental agents and study their mechanisms in which they induce disease. The information that is generated by the NTP then is used by regulatory agencies to establish guidelines, evaluate risk, and protect human health. My role at the NTP was largely involved in the design and interpretation of numerous toxicology and carcinogenicity studies. I was the principal author on several technical reports generated by the NTP on chemical substances in the environment or in occupational settings. I've also studied mechanisms in which chemicals cause disease. What is the behavior of a chemical once it enters a body? How is it absorbed, metabolized, distributed to tissues? I was also a member and for part-time chair of a NIEHS review group for the National Toxicology Program's report on carcinogens. This is a report that is mandated by Congress to determine whether agents that citizens are exposed to are possibly or likely or known human carcinogens. I think you asked me also what was my role in the cell phone study. I led the design of the NTP's carcinogenicity studies on cell phone radiation in rodents. At that time, I was the senior toxicologist in the NTP. I was the director of what was called the special programs in the environmental, at the NIEHS in the National Toxicology Program.
So can you describe the NTP cell phone study, the study itself, and more generally how it was designed, who was involved, what sort of oversight the study had?
Okay, so the design of the study was aimed at characterizing the potential toxicity and carcinogenicity of cell phone radiofrequency radiation in rats and mice of both sexes. The studies were conducted at frequencies. These are 900 megahertz for rats and 19 megahertz for mice, with modulations that are used in cell phone communications. I'm not sure if you want me to talk about what a modulation is or not. The frequency that I gave you, the 900 and 1900 megahertz, these are carrier waves. In order to carry information, that carrier wave is modulated, and there are different types of modulations on the carrier wave. This is by changing either the amplitude of the wave or the frequency of the wave with small increments. And this enables multiple people to have access to the cell phone tower. The abbreviations that are used are GSM and CDMA because people may see that on their cell phone or on the box that this is a GSM modulated phone or interacts through a GSM modulation or CDMA. So those the common modulations that are used in cell phone communications were what we used in the NTP studies. Okay, little was known about the possible health effects on the long-term exposure to what would be a non-thermal or a minimally thermal level of exposure. In fact, the guidelines that have been established by the Federal Communication Commission's, FCC, are based largely on protection from acute injury due to a thermal effect. So the NTP study was designed to evaluate the health effects, and at that time, and even still today, many people will say, or physicists anyhow, that cell phone radiation cannot cause a biological effect other than heating. And the reason they say this is that cell phone radiation is what we call non-ionizing radiation. It's not strong enough to break chemical bonds. It can't break itself bonds within DNA. So the expectation was that there would be no biological effect, especially as it relates to cancer, if animals were exposed to non-thermal levels. So the studies then were designed to test what we call the Null Hypothesis, being that there would be no effect in animals from cell phone exposure to cell phone radiation. And if we were to see an effect, we included several exposure intensities so that we could enable information to be provided to regulatory agencies on dose-response relationships. As we'll probably discuss in a short while, I think the study has clearly shown that that no effect hypothesis has now been disproved. In terms of the design of the study, this was done in 3 phases. The first was what we called a thermal pilot study, where small groups of animals were exposed to cell phone radiation at different exposure intensities so that we could identify an exposure intensity that would have a minimal effect or no effect on body temperature. And we wanted to identify the maximum exposure intensity to which an animal could be exposed that would not increase body temperature by 1 degree centigrade. In essence, no measurable effect on body temperature. The second phase was what we call a pre-chronic study, where animals were then exposed to multiple exposure intensities below that which would increase body temperature to see would there be any toxicity produced that might affect the longevity of animals in a 2-year study. And third then was the chronic 2-year study, and that's the one that's aimed at determining the chronic toxicity and carcinogenicity of cell phone radiation in laboratory animals. Now, to begin this project, my background in toxicology had been mostly chemicals, and cell phone radiation is a physical agent. We sought the help of physicists and engineers at the National Institute of Standards and Technology. They're located in Boulder, Colorado. And together we came up with the idea of using what we call a reverberation chamber. This is a scenario in which we could expose animals unrestrained to cell phone radiation. And the reason for this is that many of the previous studies that were conducted on cell phone radiation exposed animals that were in tubes so that the animal would remain in a fixed position with respect to the antenna. In a reverberation chamber, the animals could be exposed unrestrained, and this would enable us to expose them for many more hours per day than exposing them in tubes. The other agency that we worked with was ITIS, Information Technologies in Society. They are located in Zurich, Switzerland, and they worked on validating the reverberation chamber approach. They built the chambers, they computed the amount of amplifiers that would be needed based on the number of animals that would be present in the chamber. They built special chokes for the water delivery system, because if an animal is exposed while drinking water, there could be a sparking effect between their mouth and the water, which could be adverse, and they may not drink their water. So they devised a special choke such that when the animal was drinking, their mouth was in an unexposed environment. The other group were scientists from the National Toxicology Program. They worked with me on a number of details of the experimental design. And fourthly was the Illinois Institute of Technology Research Institute. This is the ITRI. This is a laboratory in Chicago, and that is where the actual animal exposure studies were conducted. I had oversight on all of these phases until I retired in January of 2009. The person responsible for this type of work is called the project officer, and I was the project officer on this contract. Compared to other studies, this was more than a simple one chemical study, because we included two modulations, which in essence is double a single chemical study. And there was a large expense involved in developing the reverberation chambers and validating that type of approach. So the cost, I believe, was in the range of $25 million, but it might have increased from the time I left until the studies are completed. So it might be somewhere slightly above $25 million is my estimate. Because of the nature of the study, the magnitude of the study, obviously the public interest, in 2003, we presented at the Bioelectromagnetic Society meeting the design of this study with descriptions of the reverberation chambers, the validation of the reverberation chambers, the number of animals that would be included in the NTP study, the endpoints, and this was quite well received at the BEMS meeting. In fact, to use a popular name right now, this statement was that this would trump all other studies that had ever been done on cell phone radiation.
Essentially, anybody who might be interested in the study would have had some opportunity to comment and give feedback at that point as to the design of the study.
Oh, yes, yes. This is all open, transparent.
Right.
We described in very much detail the experimental design and the system itself prior to even building the chambers for the study. What we presented at the BEMS meeting was the preliminary workup that had been done in Zurich on the validation of the exposures to show that the exposures were uniform within a chamber. Because if we're putting 100 animals into a single chamber, we want to be sure that all animals are receiving essentially the same exposure. Those on a top shelf should not be different from those on a bottom shelf or in the middle of a chamber as opposed to the sides. A reverberation chamber is somewhat similar to a microwave oven, but it's a room. So it's a shielded room. That means that electromagnetic fields don't penetrate the chamber from the outside in or from the inside out. And within the chamber, there is an excitation antenna, and this creates electromagnetic fields. But within the chamber, there are also paddles which are stirring these electromagnetic fields, which are then bouncing off the walls. Anything which does not absorb is reflecting these electromagnetic fields, and the paddle is metallic itself. So it's stirring the fields, and if you set it up properly, and that's what a lot of work was done, the paddles the paddles have multiple angles on them, and therefore the fields, the exposures, are coming from multiple angles, in fact, from all directions. And that was one reason why we could expose animals unrestrained, because even as the animal moves around in its cage, it is exposed from all different angles. So the paddles created what's a statistically homogeneous electromagnetic environment in which the animals are exposed. And because they are unrestrained, then they can be exposed for extended periods of time. When animals were exposed in tubes, I mentioned, I think the maximum exposure duration was 2 hours. In a reverberation chamber, we could expose them for a much more extended period of time. The NTP released some of the findings from the studies. The findings that they released were the tumor findings in rats. And what was reported was that there were an increase in brain tumors. These are gliomas, as well as malignant schwannomas. This is a rare tumor of the nerve sheath. These are schwannomas of the heart. These were observed in exposed rats, but not in controls. And there were also increases in DNA damage in brain cells of both exposed rats and mice. And probably the important point about this is that in 2011, the International Agency for Research on Cancer, IARC, classified radiofrequency radiation as a possible human carcinogen, and that was based largely on the reports of increased risk of gliomas and acoustic neuromas. The acoustic neuromas are tumors of Schwann cells of the acoustic nerve. And this was reported for long-term human users of cell phone. So the consistency in the findings of our animal studies, gliomas and schwannomas, with the human studies reported for the basis of the IARC determination of a possible human carcinogen, gliomas and acoustic neuromas, I think is rather amazing. What was shown for DNA damage were strand breaks, DNA strand breaks in brain cells. Now, as I mentioned earlier, the radiofrequency radiation does not have sufficient energy to break bonds per se. So we really don't know the exact mechanism by which the DNA strand breaks occurred. One possibility is that they occurred in relationship to another effect that might have been induced by the radiofrequency radiation. And one example for possible reasons is that there was oxidative stress. Under the conditions of oxidative stress, you can create oxidative DNA damage, and that is a possibility that might have happened in the brain cells of rats and mice, that the radiofrequency radiation may have induced oxidative stress, causing oxidative DNA damage, leading to a strand break, and that perhaps in the white blood cells, that was not the case. So a negative for micronuclei and a positive for DNA damage in brain cells can have explanations related to the mechanism in which the radiofrequency radiation is causing its effect. And that's the case where additional mechanistic information would be very useful to answer that type of question. I was involved in an IARC exercise looking at the concordance between animal and human cancer findings, as well as mechanisms of carcinogenicity. And a paper was written, co-authored first by Martin Smith. There's a number of authors. I'm not an author on that paper, but they list 10 characteristics of chemical carcinogens, human chemical carcinogens, and one of those 10 characteristics is oxidative stress. The lower birth rate, to me, indicates it is a finding. It indicates that there is a biological effect. And as I mentioned, the exposures were designed to not affect body temperature. So why that exists, I don't know, but it would be an area which I would think people would want to investigate because, again, it's an unexpected finding. I was concerned earlier than the release of the NTP data because I was a participant at the IARC meeting in 2011 on radiofrequency radiation. And in developing the project for NTP, I had to review the literature to understand the basis for this type of study in terms of animal studies, mechanistic studies, and human studies. So I began reading a fair bit of the epidemiology literature, noticing that in particular in Sweden and other countries, there were reports coming out on increases in gliomas and acoustic neuromas. So I didn't know prior to the study what would happen with the NTP evaluation in animals, but I had this concern based on knowledge I had from the epidemiology studies. And at that time, I was encouraging my children to reduce their contact with their cell phones and not hold them against their head.
Been a number of articles in the lay press criticizing the design of the study and dismissing its findings. I'd like to go over a few of those criticisms and get your response to them.
Okay.
One that's been I've seen in many places is just the common criticism of toxicology studies, that it's an animal study and doesn't represent humans and an effect in humans.
Okay, well, it's strange that that criticism is made since animals are used in the pharmaceutical industry to test drugs before they expose humans to drugs during drug development. And the reason that animals are used in the pharmaceutical industry is because they exhibit similarities in their biological processes related to disease induction. So that similarly in the toxicology arena, animals are used because in this case it's unethical to expose animals to environmental agents to test them for their toxicity or carcinogenicity. But probably even more importantly is that every known human cancer carcinogen that has been shown to cause cancer in humans has been shown to also cause cancer in animals when tested appropriately. In addition, approximately a third of the human carcinogens were first identified in animals before they were verified as human carcinogens in human studies. Equally important is that the animal studies provide great control over the exposure condition. And this is important because it eliminates the possibility of confounding factors, which often show up in human studies as being a potential contributing factor to human findings. So the animal studies have this exquisite control over the exposure condition, the avoidance of confounding factors, and the animal studies are reliable in showing carcinogenicity with all human carcinogens. So I think that correlation adds great strength to the reason for studying in animals. And from a regulatory perspective, because animals have a shortened lifespan compared to humans, information on carcinogenic potential can be determined in a much shorter time interval, as opposed to having to wait for a clinical manifestation of a disease, which in many cases may take 20 or 30 years. So I think there's many advantages of the animal model. And as I said, for the most part, disease induction is similar in animals and humans.
I understand that the animals in the study were exposed to cell phone radiation for 9 hours each day during the study. Given that humans do not tend to use a phone 9 hours a day, how do you relate the animal exposure to the human exposure?
So in this study, animals were exposed for 9 hours a day. The exposure period actually lasted 18 hours because the exposure in each chamber was on for 10 minutes and off for 10 minutes in that type of cycling. And that's where it ends up as totals 9 hours per day of exposure. But there's several reasons here. First, there's been great increase in the use of cell phones over the past 10 years. And there's also extensive use of multiple wireless devices that also emit radiofrequency radiation. So most people are actually exposed to radiofrequency radiation for much more time than the time they spend on their cell phone. 8 hours a day, 9 hours a day is probably not uncommon. And that's why I believe the daily exposures are not unreasonable. But similar to studies of toxicity or carcinogenicity in animals, what is typically done is to expose animals to higher concentrations than humans are exposed to. And this relates to the sensitivity of the bioassay. The bioassay cannot detect an effect unless there's at least a 5 or 10% increase. So in a toxicity study or carcinogenicity study, the exposure concentrations of a chemical are higher, and there is an extrapolation down to human levels. In many cases, it may be comparable of an animal study and an occupational exposure. But from the dose response information, which I mentioned the study was designed to obtain, information can be gathered in terms of what is the risk at lower exposure concentrations. So that you need to expose, we couldn't expose animals to higher intensities because of the factor of heating. So we increased the time of day to challenge the hypothesis, the null hypothesis, that there would be no effect on a biological system. And with the dose response information, agencies could characterize risk at any exposure levels. And as I mentioned, people are exposed to radiofrequency radiation from multiple devices throughout the day. So I think exposing animals for 9 hours a day is a reasonable exposure duration. I think exposing animals for 2 hours a day would probably underexpose.
Isn't it necessary to have a higher exposure in an animal study also because the animal study only lasts for a year or two, whereas humans are using these for decades?
That's true, but people argue on that in terms of, are we comparing a lifetime of an animal to a lifetime of a human? So if it's lifetime based, it's not unreasonable. However, a 2-year-old animal, and that is what's used conventionally in toxicity and carcinogenicity studies, is actually equivalent to approximately 65, 70-year-old human. And there have been arguments in terms of whether the duration of carcinogenicity studies should be extended to a full lifetime. In fact, at the Ramazzini Institute in Italy, they conduct lifetime studies as opposed to 2-year studies. But if you're comparing lifetime to lifetime, that is the basis for the comparison between animals and humans.
It has been noted that the control animals in the cell phone study didn't live as long as the exposed animals. Is that correct? And what do you make of that difference?
Well, this is a point that has been raised on several occasions. I don't believe the survival difference actually has a great impact on the results for at least 2 reasons. One is there was no statistical difference in the survival between the male rat group that had the highest incidence of gliomas and the schwannomas of the heart compared to the male rat control group. This is something which hasn't been brought out by the critics of the NTP study. And second, there were also precancerous lesions that were observed in the NTP study. The precancerous lesions in the brain were glial hyperplasias, and there were also hyperplasias in the heart of Schwann cells. No precancerous lesions of those types were seen in control animals. The glial cell hyperplasia, this is the precancerous glioma, these were detected as early as week 58 of the 2-year study, 104 weeks. And the heart schwannomas that you mentioned, these were detected as early as week 70 in exposed rats. Thus, there was sufficient survival to detect these tumors or precancerous lesions in the brain or heart of control animals. Most animal experiments for carcinogenicity include 50 animals per group. This study, we increased it to 90 animals for the 2-year study per group because we wanted to try to increase the power of the study. Increasing it to 90 animals does increase the power somewhat, but too little power is more likely to produce a false negative than a false positive. Because remember, we're testing the null hypothesis, the null hypothesis being that there is no effect. Therefore, to say that there is a statistical effect, you need to see a response which is detectable with 95% confidence. So to reject the null hypothesis, to say that the null hypothesis is not true, you need a more powerful study. Low power then would not provide enough animals to say that there is a difference. And you cannot reject the null hypothesis. So I think it's a reverse in terms of the issue of statistical power. We increased the number to increase the power, but in spite of that, in an experimental study to detect a statistical difference, such as for a rare tumor, as we're discussing in these studies, you need somewhere of 5 to 10% increase to reach statistical significance. But a 5% incidence, if this was something happening in the population of cell phone users, would be it would be a catastrophe. such that the power is low for expressing to detect a strong enough response when the numbers are relatively small. But we also have trends, and trends are important. The trend analysis says that as you increase the exposure, the response is increasing in a positive sense. So, and in many of the groups, in male rats in particular, there were statistically significant trends.
Now, it's also been noted that the fact that the control animals had no tumors was unusual and was below the rate of tumors seen in past studies for control animals. What do you make of that criticism?
Okay, well, in toxicity and carcinogenicity studies, the most appropriate control is the concurrent control as opposed to control animals from other studies. And for rare tumors, such as gliomas and schwannomas, it's not unusual to find no tumors of those types in control groups. However, as I mentioned before in describing the reverberation chamber, this is a shielded room. It's shielded also from external electromagnetic fields. So I can't say for sure, but perhaps the housing of rats in the shielded reverberation chambers affected the tumor rate of the concurrent controls, reduced them. This we don't know. Unfortunately, there's not sufficient data to answer that particular question, but no other study has been conducted in a similar environment, and for that reason, it's most important that the comparisons be made between the exposure group and that concurrent control group.
To restate that, there never has been a control animal before this study that lived in an environment where they were electromagnetically shielded for the duration of the study.
That is correct.
Similarly, it's been noted that the cancer risks, the significant cancer risks, were only seen in male animals and not female animals. And that it's been suggested that, again, that implies that this is somehow an anomalous effect, and it's not a consistent effect.
Well, so yes, there was a difference in the response between the male rats and the female rats, but I want to point out that there were gliomas and schwannomas of the heart in some of the exposed female rats. The numbers were smaller than were seen in male rats. There were also precancerous lesions in female rats that, as I mentioned, that have been seen with male rats. The numbers were smaller between female rats compared to male rats. However, this is not an uncommon observation where a difference in response is seen between male and female rats. This happens frequently in toxicity and carcinogenicity studies. But I should also point out that there are gender differences in cancer rates in humans. For example, for brain cancer, mortality rates are approximately 50% higher in men than in women. And for many human cancers, including cancers of the heart, kidney, non-Hodgkin's lymphoma, the incidence and mortality rates are higher in men than in women. So I can't explain why there was that difference, but a gender difference is not an uncommon observation in animals as well as in humans.
It's also been suggested that the fact that you see an effect in the CDMA group, but in one frequency modulation group, but not in the other, implies that this is just a spurious effect and doesn't reflect the impact of cell phone radiation. What do you think?
Well, again, it's similar to the male-female rat difference. The numbers were different, but there were brain tumors and schwannomas of the heart in both GSM and CDMA exposed groups. The numbers happen to be slightly larger in the CDMA versus the GSM, but I don't think there's been a statistical comparison between the two groups. And if there was, I'm sure they wouldn't show a difference, because there were increases in both groups compared to the control group.
What do you think the implications are of this study for public health?
Okay. Well, so as we were discussing, the findings of brain tumors, the gliomas, and malignant Schwann tumors of the heart, as well as the DNA damage in the brain cells of exposed animals, as far as I'm concerned, should raise the public health concerns to a much higher level than exists now. And this is especially true because these were tumors of the same types of cells that have been reported in human studies, that is, the gliomas and acoustic neuromas from epidemiology findings. So in my view, this new evidence supports the human cancer data and indicates to me that a higher classification, cancer classification for radiofrequency would be appropriate. IARC, in their evaluation in 2011, concluded that radiofrequency radiation is a possible human carcinogen. I think with the new data, a classification of probable human carcinogen would be more appropriate. But in addition, there's a concern for children, because for children, they may be at a greater risk than adults, and this is due to the greater penetration and absorption of cell phone radiation in the brains of children, and because the nervous system of children are more susceptible to agents that cause tissue damage. So when there are findings in adults, the concern level for children would be even greater than just the adult human data.
Is there sufficient information and data at this point to cause should the government be introducing new regulations concerning the design, manufacture, and use of cell phones and related devices as a consequence of this information?
Yes, I think very much so, that with this new information, I think the regulatory agencies need to make much stronger recommendations to consumers. And this recommendations in terms of taking precautionary measures to avoid close contact with cell phones, and especially the use of cell phones by children. Now, the current recommendation is by the FCC, the Federal Communication Commission, is an exposure limit for cell phones of 1.6 watts per kilogram, averaged over any 1 gram of tissue. This was a exposure limit that was established almost 20 years ago to avoid acute injury due to heating effects. The exposure intensities that were used in the NTP study were not different from that 1.6 watt per kilogram. The exposure intensities were 1.5 watt per kilogram, 3 watt per kilogram, and 6 watts per kilogram. These are not very different from the exposures that cell phone users might experience. So in my view, the FCC needs to revisit this exposure limit and work with the FDA, who is interested in this type of work, to quantify the cancer risks associated with the use of cell phones or other wireless devices. And the manufacturers, I think, need to redesign cell phones and other wireless devices so that they provide new devices with much lower emissions of radiofrequency radiation. Until
there are more appropriate regulations, what can you suggest that people using cell phones do now to protect themselves and their children?
Well, the recommendation that is made by FCC is take precautions if you are concerned. I think that recommendation needs to be changed to take precautions. People need to take precautionary measures to avoid close contact with their cell phones. Use a speaker setting, use headsets, text whenever possible, but not when driving, and especially avoid use of cell phones by children. People should be aware that cell phones and wireless devices are not toys, and these are something that children should not be playing with. And people might now start considering reducing their reliance on wireless devices in their homes, and perhaps as well in schools. Probably the greatest exposure to radiofrequency radiation is through cell phones, especially for people who hold the cell phones next to their heads. But there are, in addition, other sources. All of our wireless devices are based on radiofrequency radiation. So your computer that's wireless linked to the router in your house is emitting radiofrequency radiation. Your tablet is also connected through radiofrequency radiation. In these devices, there is an antenna which is emitting radiofrequency radiation. Cordless telephones also emit radiofrequency radiation. So there are these common devices which people use on a regular basis that are emitting cell phone or emitting radiofrequency radiation. And of course, the router in the house. There are monitoring devices that people use for their children, which are also based on radiofrequency radiation. So there are numerous devices, and I think the number of devices are increasing because there's a convenience factor associated with these kinds of communication devices. That people do not want to throw their cell phones away, and the government is not going to ban cell phones. So the next step is, what's the best way to reduce exposure? As I mentioned, when the dose response data are evaluated, there will be estimates of risk that can be derived from that dose response information and compared with the human data. And in chemicals in the environment, the risk that is typically accepted is 1 per 100,000 or 1 per million over lifetime exposure. However, with the cell phones, I'm sure the risk is going to be much greater than 1 per 100,000, with the exposure limit that is now established for thermal effects. This is the 1.6 watt per kilogram. If the FCC lowers that, it puts the burden then on the cell phone manufacturers to come up with a product in which that exposure is reduced substantially. And this is something which I feel needs to be done. And I think it has been done to some extent. So for example, on the cell phone device, the antenna used to be at the top of the cell phone, which would be right next to the head. They've moved that antenna towards the mouthpiece because they're aware of this issue that they need to reduce the exposure to the brain, because as you increase the distance of the antenna, the exposure intensity decreases by that distance squared. So there are ways in which you can hold the cell phone away from the body, and this will one approach. I believe even the cell phone manufacturers describe a certain distance in which the cell phone should be held away from the body, even when not communicating, because by voice, because the cell phone is communicating with the cell tower even when you're not using it for communication. So can the devices be adjusted so that a clear signal can be derived with lower emission intensities? From what I've heard, yes, that is possible, but there hasn't been enough energy to deliver such a device, and hopefully the new data will provide that push in that direction. Don't treat your cell phone or wireless device as a toy. Considering it is a device which is emitting radio frequency radiation, and whereas the past people thought that that could not create an effect in animals or humans or any biological tissue, we now know that that is not the case, and the findings, the reports need to be taken seriously, because you don't want to find out later that a precautionary measure which could have been taken wasn't taken. Better to be safe than sorry.